and to go at why it's important to repair the DNA damage and what are the diseases or things that can happen when we have too much DNA damage. And I think I sent the shortened presentation. I will send the full one when we finish the class because I think I sent like half of it. But we'll see how much we can reach. So again, stop me whenever you need. And we will stop with the very basics and then we'll go into the mechanisms of DNA repair. So first of all, a mutation and DNA damage, they are not the same. A mutation is already a change in a base that is not recognized as something is wrong with the genome, right? It's already inside the genome and cannot be recognized as something that was wrong. DNA damage creates a disruptor in the DNA. It can be a bulky adduct, it can be a broken DNA, so it's different. But of course, from DNA damage, we can generate mutations because when it's not repaired correctly, we can have new mutations. Okay, so what are these sources of mutations and DNA damage? We can have all the exogenous reasons, such as oxidative damage or depurination that occur normally because the chemistry of the bases. And we can have radiation and chemical adducts that are also from the sunlight or from chemotherapy or any other reason or contaminants that can lead to DNA damage, like in cigars or all these things. They mainly lead to DNA damage, that this is one of the main causes of cancer. And we have also polymerase errors. These are endogenous. And we have error-prone DNA repair, that we will get to that. So we can have different types of damage. In here, for example, we can lose a base, a purinic site. Usually we lose a base, still the phosphates and the sugar are connected, but here we're missing something. We can have the amination, like here, where we lose a part of the base, and then it's uracil, okay? And then this is not the correct one, it's not the correct base pairing. Then we can have mismatch, and this usually occurs because polymerase makes some errors, and we have a mismatch repair error. We can have double-strand breaks, and these are the most dangerous types of damage because we can lose genetic material, and also we can link together wrongly, for example, two different chromosomes or parts that were not supposed to be together. So not in cell lines, but in mature cells, a single persistent double-strand break can send the cells to apoptosis. So this is very dangerous type of damage, and it is the one that has more signaling and alerts the cell the most because it cannot tolerate this. We can have intercalating agents, and most of the cancer drugs that we use today are these kind of intercalating agents that disrupt our DNA and try to send the cell to kill itself, to apoptosis. Single-strand breaks and bulky adducts that can be oxidation or other types of things. So it can happen every day. The estimates are a little bit weird because it's from 1,000 to 1,000,000 lesions per cell per day. Of course, it depends on the cell. If we are talking about skin and we are under the sun, we have many more lesions per day than in a different tissue. But we have at least 1,000 different lesions per cell per day that we have to repair. And depending on the kind of lesion is how much the cell will have to activate all these downstream signalings to allow the cell to cope with the DNA damage. And we have many different types of responses and outcomes. So what are the different repair pathways? And first we'll start with the very basics that got the Nobel Prize to three important researchers in 2015. Thomas Lindahl, that he discovered BER, the base excision repair. And the first thing that he thought is that with the rate of irradiation and damage that can occur to the cells, life would be impossible on Earth if we wouldn't have a repair mechanism. No? This will be incompatible with life. So there has to be a mechanism, and he found this BER mechanism, that we'll talk about it. Aziz and Carr that discovered the NER, nucleotide excision repair, and we'll see the difference between base excision repair and nucleotide excision repair. It sounds almost the same, but it's not the same. We'll talk about it. And he found that in the bacteria there is a light system that photolyases, using the light, the photons can activate a repair mechanism, but this is lost in mammals. And he discovered also a dark system that can repair DNA damage without this photoactivation. And Paul Modrich that discovered the mismatch-based repair. That again, in some systems, he discovered that to identify which one is the correct, because when we have a mismatch, A and a G, for example, which one was the correct one? How can we know which one is the correct strand? So he discovered that in some strains, there is a methylation in the paternal or the maternal RNA, that when the polymerase went through, it keeps the methylation, and the new one doesn't have the methylation, so the original one is the one with the methylation. Now, we don't have this system anymore. It's not true for mammals that the methylation is retained only in the maternal DNA, and we are not completely sure how our system managed to identify to which base should we do the repair. So in many cases, there are also mistakes, but there is also an open question. Okay, so these are the Nobel Prize winners for 2015, just to point out how important DNA damage and DNA repair are. And we will start with the base excision repair, that it corrects very small base lesions, like one single base that is oxidized, that is deaminated, that is missing. This is the system that will correct it. And it has 4 steps. And in here we go a little bit into biochemistry. I hope you are still with me, but it's just to give you a sense of how things work. And of course, this is not very important to memorize or remember all these details. So we have here the amination, and this is incorrect. So the first enzyme that recognizes can cut only the base, but leave intact the phosphate sugar bonds. But then we need to add a new base, so we have to open a link, a nick in here. A polymerase will add the missing base, and then we have to ligate this one, and then we are done. And this kind of damage occurs a lot in the cells, but it does not activate a full DNA damage response. It's like we can cope with this. This is a normal type of damage that we can continue with cell division and any other things. We can have also a short patch and a long patch, meaning that if it's more than one base, it's like 3, we will use a different polymerase, and then we have extra step where we have to cut these extra bases. We have like a triple strand right now, and then we have to cut these ones and get rid of them. So this will be a long patch there, and this will be a short patch there. And the difference is not very important. It's just the polymerase that arrives to the site of damage, and if it will get help from this FEN1 pCNA. So we have this short patch and long patch ways of activation. And what I want to give you is just an example of how BER can be affected as we age, or how it can cause disease in the long term. Because this is one of the most common types of damage, is oxidative damage what causes most of the BER damage. There was some research trying to identify how this affects as we age. And what they did was transcriptomics. They took all the RNA from different brains from age 26 to 106, I think, and just saw what is changing in a continuous way with age. And some of the top categories that were changed were the DNA repair system, and downregulation were synaptic activity and things that are important for brain functioning. So as we get older, we have more DNA damage, but we have less capacity to cope with this oxidation and with this DNA damage. So what happens with oxidation is usually we get a deoxyguanosine that can be converted to this 5-oxo-deoxyguanidine. This is a very common mark to find when we have DNA damage. It is in the genome, and we also have antibodies to identify it. So it did give us a tool to investigate how much of this we have in every tissue. And what they did, they developed a very intelligent system, in my opinion. So what they did is they took fetal DNA that is not damaged, or fetal DNA that they add H2O2 to allow damage. Now we have the same DNA with oxidation and without oxidation, just as a proof of concept to see if we can measure how much oxidation we have. And then there is this restriction enzyme, FPG, that recognizes and cuts only the oxidized DNA. It recognizes that something is wrong and cuts this oxidized DNA. So as a proof of concept, they took the fetal, they measure how much intact DNA we have, and after oxidation, we have less intact DNA of several genes. So this is our control that the experiment is working. Any of you work in molecular biology, or most of you are computational? Yeah? Okay. So we always have to do many controls to see that the experiment that we invented is working. And then they took samples from this 26-year-old and did qPCR. Do you know how the qPCR works? Most of you? No? Okay. So you know how PCR works? Okay. So what we have is that we have every cycle, if we have one molecule, then we will have 2 molecules, 4 molecules, and when we see an exponential growth, and then we get to a plateau because the system cannot work anymore, the enzymes are not so sensitive or anything. What we do is we add a fluorescent molecule that can intercalate only in double-strand DNA. So each time that I have more molecules, I have more fluorescence. So I can measure the fluorescence in time to see how much DNA or RNA I have originally. So for example, if we go up in cycle 20, we have more than if we go up in cycle 30, right? So here I have more DNA because it came up earlier than if it came up here. So this is what they did in here. They said, okay, let's measure how much DNA we have with the 26-year-old in this gene with FPG cleavage. And the difference was not so big. But then if we compare the same gene in a 77-year-old, we see that this is severely delayed, right? We have much less intact DNA. The DNA is damaged in these older samples. And they measured various genes, and they saw that this was happening for all of them. And also, we can measure with antibodies how much of this 8-oxo-deguanidine we have, and we have a huge increase. So as we get older, our brain accumulates this kind of damage, and this kind of damage, in the end of the day, can affect the polymerase activity or can change the gene sequence, right? Because if we don't have a correct base, we could have kind of mutations in the mRNA level or when the base pairing occurs. So this FPG enzyme, this is what I explained you. I forgot that I have the slide with explanation, but this is how the qPCR works. The PCR will amplify, and the enzyme will cut the DNA that is marked. And another thing that we have been seeing is that the enzymes that are in charge of Bair repair, like OGG and AP1, they are decreased in animals, also in the brain, but in any other tissue. So as we age, we have more accumulated damage because it happened spontaneously, but our capacity to repair it goes down. Why it goes down? One of the things that they saw in this nice paper is that when the regions are open, because these are genes that we need to use, we have more chances of get them damaged, get them oxidized and everything. And then they become impaired because they cannot be transcribed because they have damage. So then we have less of these DNA repair enzymes, and then we have this cycle where we have more damage, but we are not able to repair it, and in the end of the day, this causes tissue damage. Okay, so to summarize Bair, we have a single base that is damaged, that is recognized by a glycosylase, that it flips the DNA a little bit to put it towards the outside. Then we generate a site without any base. Some of the enzymes have double activity, like APE is the one that generates the nick, but some of these enzymes can also generate a nick by themselves. And then we call the polymerase to add a base, the ligase to close it, and then we are done. We have repaired the DNA, and we didn't cause more damage. The DNA is okay again, and we can continue with our lives. Now, why is also this very important for the epigenetic regulation? Because methylation in the DNA, it's a very unfavorable reaction to reverse. If we have a methylation, we have to invest so much energy to do demethylation that this doesn't happen. There is no enzyme that can do demethylation. So the theories were that to remove methylation, we have to go through passive dilution. So each time the DNA divides, we have less methylation. And then it was discovered that we can modify the methylation with some enzymes that I'll show you soon, and use the DNA repair system to induce demethylation. Because at the beginning, when the methylation was discovered, it was thought, okay, this methylation remains there forever. If the DNA is methylated, this is an epigenetic modification that we cannot remove. But then it was discovered that we can change it slowly to remove it completely. So what happens is when we have the 5-cytosine methylation, that is the most common one, and usually is in CpG islands. The promoters have this kind of CpG-rich regions. Then when we want to silence, we methylate these regions, and then they remain very close. So we have a methylation that we cannot remove easily, but then we can start using TET enzymes that lead to oxidation. So 5-methylcytosine becomes 5-hydroxymethylcytosine, 5-formylcytosine, and 5-carboxylcytosine. And then this is already recognized as DNA damage. Then the bare enzymes that I was telling you about, they recognize this that was done on purpose on the genome as DNA damage. So they remove it, and they add the correct base without methylation. So we can use the bare repair system to prevent methylation or to do demethylation in an active way. And the second pathway uses the amination with AID and ApoBec enzymes. These enzymes can also do RNA editing. This is very common in reptiles and amoeba and other systems where they don't have so much alternative splicing, but the way to generate novel genes is to edit the RNA and then get many different variants of a single gene with RNA editing. But this can also be used. Some of these members can change the DNA, and then again, in the end of the day, signal to the bare repair system that something is wrong with this, and then we can get a non-methylated base. And this is one way to remove the methylations from the genome. The second system is called NER, Nucleotide Excision Repair. And this will recognize not a single base that is wrong, but some bulky adduct, something that looks weird in the DNA, like intercalating agents, for example, that generate like more space or change the looping of the DNA. They can recognize also photoproducts. For example, if we have irradiation and we have thymidine dimers or 6 to 4 photoproducts, the two bases are cross-linked together and are disturbing how the DNA looks in the bulk, then we will have this repair. And for example, cisplatin and other chemicals, these are the most commonly used in cancer to induce damage and send the cells to signaling to kill themselves. So there are two main pathways for nucleotide excision repair, and it's very, very interesting because one of them gives us a disease that is xeroderma pigmentosa, meaning that these kids are super sensitive to light. They cannot see daylight. They completely become with like tan and with speckles, and they get a lot of skin cancer if one of these pathways is impaired. And the other pathway, if it's impaired, we get cocaine syndrome. That is, again, a progeria, a premature aging phenotype. So we have two sub-pathways, the global genome NER, that it will repair the DNA everywhere where there is no transcription. So this is the main pathway for all the intergenic regions and all the genome that is over there. But if we have a region with genes that is transcribed, then the recognition will be different and will be the transcription coupled NER. So we'll first go to the global genome NER, that it will recognize everything globally. And this is a picture of a kid with xeroderma pigmentosa. And the thing is that we can have mutations in several genes that are part of this pathway. And any of these will be complementary because we don't manage to repair correctly to get xeroderma pigmentosa. So what is happening in here? We have to recognize the sites that are damaged. Then we have to cut a larger portion of the DNA, but larger I mean 20 base pairs and not hundreds of bases. Then we have to synthesize again, ligate, and as in base excision repair. Another thing that this system may recognize is different conformation of the genome. We have many regulatory sequences in the genome that are non-bDNA motifs, like this cruciform or the triplex or some G-rich regions that form these very big and stable structures in the DNA and are very difficult to open. Sometimes also our nerve system can recognize them and remove them. These regions have their own important roles. They can activate promoters and they can be regulatory elements, but they are very stable and very difficult to remove. So also the nerve system can use these regions to repair them, although there is no damage, and in this way remove them. And by doing so, we generate hotspots for damage. So this is one of the topics that we don't know if damage is randomly generated, if there are hotspots, but we know that some of these regions can generate these hotspots. Was there a question? No? Okay. So what happened in here is that we have a very bulky change in our DNA, and then this enzyme Qpcr complex will recognize the DNA, and then they will bring TF2H complex, that this is a bigger complex that can interact with them, and it brings some enzymes that can cut the DNA and do a nicking. But what will happen is that until all these complexes form, we won't cut the DNA, because we go from a modification on the DNA to a nick, and nicks are always worse because they can generate much more damage. So first we make sure that we have all the enzymes in place, and then we cut, and then we remove this piece of DNA that we don't need, bring the polymerase, ligase, and end this thing. So this was the one that is recognized in the whole genome, and when we cannot repair in this way, we get cancer, xeroderma pigmentosa. The second one is a TCNR, and uses these two enzymes, CSA and CSB, cocaine syndrome A and cocaine syndrome B proteins, that are also helicases and chromatin remodelers. And this is cocaine syndrome. It's very similar in the phenotype to the laminopathy I showed you yesterday. So we have premature aging when we don't manage, again, to repair our DNA. And what happens with cocaine syndrome is that they have growth development failure. They have a microcephaly, and they have a very, again, peculiar face that is smaller and very thin and with deep sunken eyes. And in this case, they do have progressive neurodegeneration, because this system is important in the whole body, and not like lamin A that is only in other tissue. This is important in the brain. So when we don't have repair system, we get neurodegeneration. And ataxia and many other things like hearing loss and mental retardation and cataracts. And it's a very, very severe syndrome. And what is different is the way of recognizing our broken DNA. If we have a global genome, then we had XPC and RAT23. But if we are in a transcribed region, we will have polymerase that will be followed by these two proteins. ERCC is the other name for CSA and CSB. Many times in biology, it depends on two people discover them at the same time. They will have two names, and it depends who you like better, so you name them. But these are the same proteins. And then after that, the same machinery is recruited, TFH2, and all the enzymes that allow the polymerase and the ligase and all the other things. So the only thing that is different is which regions are recognized and by whom. And the one is in global genome and the other one is in transcribed genes. But why do we have a so different phenotype? One of them is cancer and one of them is premature aging. Just by having it in different regions is not completely understood. So we are also trying to figure out why is this so different. Because if we go deep, if we have mutations in the downstream pathway, we will have a combination of both. But if we have only one of them, as I was showing you before, then we have only progeria or only cancer. So it's very complicated, and we think that this is because in the transcribed genes, we are generating constant damage, but we are also preventing transcription of important genes that should be expressed in there. And also we are generating, the polymerase is getting stuck, and then this may develop more problems, like from nucleotide excision repair to double-strand breaks. Because it gets stuck, it has to be removed, and it comes back. We have a collapse of various polymerases, and then double-strand breaks already send us to a very bad signaling pathway that may kill the cell. So this is the main difference in this pathway. And then we will go a little bit deeper into double-strand breaks. So before we go there, let me know if you have any questions about the other systems. Okay. So, yeah, sure. Where in the microphone?
I find it very interesting how, I guess this is a more general question in a sense, but how do you manage the specificity of finding the, because the thing is that these proteins are being produced, these DNA repair proteins, and they're diffusing around the nucleus, right?
Right.
And they're just thrown around there. And there's a specificity, which is you have to find that particular plate where the DNA was damaged.
Yeah.
And I guess you also have to avoid, because since they act in these, they form these complexes, you have to avoid forming the complex when you're not already at the site, right?
Right.
These things only should couple once they have attached to the particular site where they're going to act. So how much energy does our cells spend just producing a lot of these proteins and they go nowhere? Because it seems like a very complicated, like a very random task, right? You're just shooting into the dark, it feels to me. How much option does the cell have?
So we have a lot of these enzymes all the time, but after we get an insult, for example, irradiation or after the bombings in Hiroshima, the levels of these enzymes went up, or if we have a constant stress, they will go up. And then after the stress is gone, they will go down, but we have very high basal levels of these enzymes. What we think is happening is that they are constantly probing the genome. They bind, unbound, because if not, they will never find the damage. And this is very inefficient, right? So there are some other theories that they are trying to track them with single molecule techniques to see how they move. So they see a Brownian movement as well. But also, when there is damage, they see that they attach stronger. So one of the things that will happen, and we'll see in one of the systems that we use, is that in the first step, we have a very mild binding, not enough to activate things. So probe and leave, probe and leave. But then, if something is wrong, the affinity increases. And when the affinity increases, they can also start recruiting other proteins that can bind to them. But until this affinity doesn't increase, the other proteins cannot start forming these big complexes. And this will be the threshold to activate the downstream signaling. But we can, I will show you a system where we can fake DNA damage without damage by just persistently leaving a DNA repair enzyme at the site that we want. So the cells start understanding, okay, they are sitting here for a long time. This means DNA damage. But there is also some theories that there are some microtubulin and not microtubulin, sorry, actin filaments inside the nucleus that can move things around, right? But how exactly they bring it to the damage when there is damage is not known. And at least for example, for big topological changes, for example, a single strand break or a double strand break, they uncoil the DNA, right? They form a topological change that many things jump out and then they have to come back. And this big topological change is more easy for us to understand that something will come back with higher affinity, and I'll show you our results from the lab in this double strand break. But many of these require like a lot of biophysicists that are coming into the field, like yourself, to help us solve these problems of how they move at a single molecule, when they bind tighter, and what changes in these dynamics, because it's not completely understood. For now, we think they are kind of floating there and come and leave, but we really don't know. Okay, any more questions about this? Yeah, no, okay. So we'll go to the double strand breaks. So this is one of the things that we do in my lab. And we have many things that can lead to double strand breaks, as irradiation and chemicals and the free radicals, or any other type of damage that was not correctly repaired, can evolve into a double-strand break. Like if we didn't invest at the beginning in repairing a base that was damaged, and then we didn't guide it correctly, then it can evolve a single-strand break, and then if we have bad luck, it can evolve to a double-strand break, and this can happen and complicate things. But also we can use the specialized double-strand breaks, for example, for BDG recombination. That is when we want to generate these antibodies that we want to cut and put together different sequences to generate variability. Also, when the transposons wants to get in, they cut and get in, right? So also these transposons can induce damage that has to be repaired by double-strand break machinery. And also, in somatic hypermutation in meiosis, when we want to generate more variability during meiosis, to generate some recombination of things, we have also double-strand breaks that have to be repaired. If things go wrong, then we can go to apoptosis if we don't manage to repair, because we don't want to continue with genomes that are unstable, that can lead to mainly to cancer. That is one of the biggest problems in aging or any organism that lives more than 20, 30 years. We start increasing our chances of getting cancer. So we have two main pathways for double-strand break repairs: a non-homologous end joining and homologous recombination. And again, this is just for you to get a sense of the things. I'm sure you don't remember all these names, but just to understand in the broad terms what is happening when we have damage. So we have a system that is, let's just put together the DNA that was broken. Let's try to fix it as fast as possible. But in this way, we can lose part of the genetic material. We can generate deletions, addition, mutations. We can do a mess, but it's very fast. So if it's we have too much damage, or if we have a region that is less important, maybe we can just stick it together. And then we have the homologous recombination, that this is a more slow process that tries to find a homologous region. If there is a gene that is very similar, if we have the other chromosome, if we have repetitive elements that we can repair using them, then it will try to use the other sequence to copy what is missing and then go back to what it was. The limitation of this system is that we have to have a gene that is similar enough, close enough. So usually this happens mainly during the S phase and G2, because the chromosomes are aligned. We are just copying our own chromosomes, and then we align them also for cell division, so we have the capacity to have these genes. Or in sequences that are very repetitive, like I was telling you yesterday, the ribosomal genes, right? We have many, many different copies. So inside the nucleolus, we can all the time use homologous recombination, because we have genes that look very similar to the ones that we need. Yeah.
But during the S phase, when they are aligned, aren't they already coiled and
in S phase, not yet. In S phase, we have many replication forks that start independent in these origins of replication, and then we are starting having this duplication. Then when we are getting close to mitosis, to the real cell division, to anaphase, metaphase, all these phases, then they start coiling. So during the S phase, we are still synthesizing, and then under G2, we start compacting them and also organizing them, but still we can have some repair. But then in M, that's it. We cannot repair, and usually if we have damage at that point, the cells can go to apoptosis. But there are other things that can change the decision whether we are coming to non-homologous and joining on homologous recombination, and these are being investigated right now, because maybe if we are in a gene-rich region, and maybe this gene is important, if I just fix it with non-homologous and joining, I may cause more damage. So some of the epigenetic signals of this is an active region can maybe prevent repair from non-homologous and joining, and allow later on, when the time is correct, to repair. So we will have a slower kind of repair if the region is important. But we are not sure that this is really happening. We think it may happen, and we are trying to find out what are the signals for this, for example. So again, we need the same things that we need before, but in a more complicated way. Do you have a question? No? So we have to recognize the broken DNA. We have to remove part of what is next to the adjacent region, nucleosomes and pieces of DNA that disturb us. For example, in non-homologous enjoining, let's say the DNA is broken this way, and here it's broken also this way. So we cannot ligate because we have this, but this we are missing a part. So we will cut a little bit of this DNA until we find micro-homology. It will be enough that I have A here and a T here, and this micro-homology will be enough. But still I have to remove this little piece of DNA from here and a little bit from here to allow this to come together. So we have to do some resection of the DNA in non-homologous enjoining. And in homologous recombination, we have our DNA, and then we will cut kilobases to allow later on this strand to go to the homologous one and base pair with it to allow copying what was missing from the other strand. So we have more complicated process as the DNA damage gets more complicated. And then we have all the synthesis of the DNA and the loop and annealing and ligation. And if we copy, for example, if we use our mom gene for the gene that was from our dad, and they are different, they have different polymorphisms, then we have to activate the mismatch repair. And then we may have one or the other, depending on how we solve this issue. So when we have DNA damage, we have many, many syndromes. When we don't have DNA repair from double-strand breaks, we have usually, if it's in development, we have microcephaly. The brain doesn't manage to develop normally, and many layers are missing. This is Nijmeh breakage syndrome. That is one of the enzymes involved in homologous recombination. They have a short stature. They have immunodeficiency. As we said, we need this to form the immune system. So many of the double-strand break repairs will have also a lot of problems with the immune system and with the development of the brain. Of course, they have radiation sensitivity. We don't manage to repair damage, and if we get damage, we are more sensitive. And they get also ataxia, ataxia telangiectasia-like. This is another syndrome. We have also ataxia telangiectasia-like disorder that is very, very similar. So we have aging problems, and we have neurodevelopmental problems, and we have sensitivity. So this is telling us, as we said at the beginning of the class, that if we didn't have a repair system, this is incompatible with life. We cannot survive, we or any other organism, if we don't manage to repair our DNA. And if we have mutations that don't allow us to repair our DNA, we will have very, very severe syndromes. So what happens this is more the scheme for the cell cycle. In our decision to go to homologous recombination or non-homologous enjoining, if we are in G1, we will usually go to the non-homologous enjoining and just link the DNA together. And if we are in the S phase and getting into G2, then we'll try to go to homologous recombination. And here are the enzymes that I told you yesterday, BRCA1 and BRCA2 is later on in here, that help us repair double-strand breaks. So again, by having a mutation in these enzymes, we can, if it's one of the downstream effectors like here, we have higher risk of develop cancer. For example, if we have one that is non-mutated, one allele that is mutated and one that is not mutated, so we are waiting for the second hit. But if we have an impairment in these very important sensors, then we will start developing the disease from development and really early on and not in adulthood. So the Q complex is a complex that uses Q70, Q80, and they look like a ring, so they can hug the DNA. You see here inside the DNA, so they can hug the DNA and contain it so it won't get lost and move. And then they recruit a kinase, and this kinase is DNA PKC. because we know that phosphorylation is one of the fastest ways to signal downstream, right? So we sense that there is DNA damage, then we recruit a kinase. This kinase spreads phosphorylation to hundreds of targets, and then we activate the DNA damage response. So it looks more or less like this. It's bound in both sides, and then it's bound together to prevent losing the other sequence that we want to bind it to. And then we manage to use the ligases and the microhomology and ligases and everything to allow the DNA repair. Non-homologous enjoining is critical for this lymphocyte development. So we will cut the DNA, but of course in this case we don't need homology, right? We are generating a new sequence and we don't need the other pathway, the homologous recombination. We just want to put together the fragment that we took out back again and generate this sequence for antibody variability. So if we have mutations in all our enzymes like DNA PK, ligase 4, Artemis, or Sternonus, we will have syndromes that have immunodeficiencies, and together with other complications such as microcephaly and SCID is also an immune deficiency that we have no immune system. The other system that we use is the homologous recombination, and part of what we have to do is first of all protect our single-strand DNA. The cell doesn't like to see single-strand DNA ever. So the first that we do is we bring a protein, RPA, that binds all the single-strand DNA so it don't start reacting with things around it. And then we bring the enzymes that do the resection, cut the DNA, protecting the ends, and then helping with this attack on the other strand to allow homologous recombination and copying the other sequence. But the sensors, the ones that recognize this, are the mRNA complex, we'll talk about it, the Q complex, or CIR6, that we will talk about this in a few slides. But what I wanted to show you is a very nice molecular mechanism to how we can do this triplex and find the homologous strand. So this protein, RAD51, can bind a single-strand DNA, and let's say only one base per time. So we form a complex that has many, many of these RAD molecules, and then it can bind to a double-strand DNA and see if it has homology with one of them, and then we kick out the one that we don't need to allow copying that strand. So imagine that we have a region that is A, D, D, C, C, and then we we bring together our sequence, and we have to prove that this A matches with this one. And here we have the T because it was a double strand, T, A, G, G. So RAD comes and binds this one and check if it fits. And then there is another molecule here, another one here, another one here, checking that it fits. If these ones don't fit, then we don't have a nice homology, so we keep searching. We keep searching until we have enough molecules of this RAD51 to make it like flat, nice, all of these are bound, so we are ready to go. So in this way, we search for the other strand to find a perfect fit and copy from it. So I think it's a very elegant molecular solution to find homology one base pair at a time. And in the end, we bring some other enzymes that help us prevent more damage, like coesins that link the strands together, allowing us to copy it and not lose other proteins. And then we have to resolve this. In yeast, for example, the DNA, when it's broken, it is moved to the periphery to be repaired in the periphery. So they have hubs of repair, but the genome is much smaller. So when the DNA is broken, everything moves to the periphery where there is less action, less polymerases, less of everything, and it's repairing there. In mammalian cells, it is so big and the nucleus is much more larger that we cannot do that. So we have to immobilize it at the site where it is without allowing polymerases and other things to come and affect the DNA repair process. So what happens is that we stop transcription in adjacent genes. So if we have DNA damage in here, we will send a signal to all the genes that are over here, that if they were active, to stop producing anything. Everything in the vicinity is stopped, silenced, and cannot be used until we give the signal that they can continue. So by having DNA damage, again, we are affecting all the region and all the transcription that is next to the site that was damaged. Now we'll get to the part of the things that we do in my lab, that is DNA repair. So we work with a group of enzymes that are called sirtuins. These sirtuins are conserved from yeast to mammals, and they were first discovered because they can deacetylate histones, removing this epigenetic mark and compacting the DNA. So they were thought to be silencers, and in mammals we have 7 sirtuins. And what they noticed is that also in yeast, that they don't have the sirtuins, they live less, they have chronological aging, and if they have extra copies, they can live more passages and have less damage. Because I don't know if you know how yeast divide. Every time a yeast divides, it also releases a ribosomal RNA kind of plasmid inside the cytoplasm. So every time it divides, it's counting how many times divided by having these small RNA molecules. If we have these sirtuins, we prevent the release of many of these the circles, and then we are kind of cheating in the counting. We have divided less because we have less of these circles. In mammalians, it's completely different, but this is a way the yeast can count how they age. And if we don't have sirtuins in mammals, we have this progeroid syndrome. We have a premature aging. So in my lab, we mainly work with SIR6. And what happens when we delete SIR6 from the full body of the mouse, these mice get really, really sick. They are a little bit smaller. They have osteopenia, colitis, lordokyphosis, that is the curvature of the back. They have graying hair. They don't have subcutaneous fat. So by the age of 3-4 weeks, that is really, really young for a mice, they should live 3 years more or less, they look really, really old. So this is telling us, okay, this is very similar to all the progeria syndromes that we know. So it must be also involved in DNA repair, in addition to the epigenetic role. So what my mentor in Harvard saw is that they have more genomic instability. These are, each chromosome should be one color, and you see that there are some more combined things in the knockout. And when I was doing my postdoc, what I discovered is that when there is a double strand break, SIR6 comes really fast to the sites of damage, and it brings chromatin remodelers to allow opening of the chromatin, and now we can recruit all the machinery to allow repair. SIR6 cannot do ligation or cannot do polymerization of new strand, but it comes really, really fast and allows the opening of chromatin. But we saw that it was coming super fast. By 5 seconds, this is the laser-induced damage that we can do. We take cells, we put SIR6 with RFP fluorescent protein, and then we use a laser that can generate damage in here. And you can see here that SIR6 start arriving to the sites of damage really, really fast. So we can track the movement of proteins to the sites of damage with this method. And we thought, okay, who is recruiting SIR6 to the sites of damage? It's coming so fast, what is happening? And again, to initiate the DNA damage response, first we have the sensors that recognize that something was broken. Then we have these kinases that will spread to the cell that something is wrong. And then we'll have the effectors that will activate the cellular response. For example, if we were replicating, we should stop replicating. If we have need to remodel chromatin to allow repair, we have to do that. But also, for example, DNA is our main molecule that we don't have many of it. Proteins, we can generate new proteins. Mitochondria, we can generate new mitochondria. But if we damage our DNA, we lose this information. So all this cell will stop whatever it's doing, including energetic activities, to repair the DNA. So we can stop some of the cellular metabolism and spending energy until we repair our DNA. And if it's too much, we will go through senescence or apoptosis. If we fail to do that and the cell keeps dividing, then we'll get cancer. So these are the different outcomes that we can have when we have a double strand break. So we have the sensors that arrive within seconds, and what was known is that we have the MR11 complex, the Q complex, and PARP, that within seconds they are there. So we said, okay, let's silence them or inhibit their activity with different chemicals or by shRNA or siRNA. And you can see here SIR6 is in GFP, is in green, and here we have the control, the macro domain, that it arrives to the sites of damage very fast when it recognizes pyrylation, that is done by PARP. That is a chemical modification. And when we silence PARP or we inhibit its activity, SIR6 come anyway to the site of damage, but the control cannot come. So we know that the experiment is working, but SIR6 couldn't care less. Then we silence the Q80 or MR11 from the different complexes. This is from the non-homologous enjoining, and this is from homologous recombination complex. These are the sensors. And again, their own control, Q70 cannot come, NBS is impaired, but SIR6 comes anyway, and in some cases even better. So SIR6 was independent for any other sensor that was known. So next, we decided to see if signaling could be important. And for this, we used the system that I was telling you about to fake a DNA damage. So what we do is take a LAC repressor domain. These ones bind really, really tightly to the DNA. And we put, for example, a DNA repair enzyme, such as ATM is one of the kinases that spreads this signaling, and add a fluorescent molecule so we can track it in the cell. So you can see here in the cell we have a single sequence with 256 repeats of LAC. So we have enough molecules that are fluorescent to see a big dot, like you see in here. And we measure if we have phosphorylation of H2AX. That is our main epigenetic modification signaling that we have DNA damage. So when we put ATM and we have phosphorylation of gamma H2AX, but SIR6 doesn't come. And as a control, of course, we have some protein that is completely unrelated to DNA damage, just LAC with GFP, and this does not activate any signaling and also doesn't bring SIR6. So in this way, we are faking a DNA damage. We are allowing signaling, but signaling by itself cannot bring SIR6 to the sites of damage. So maybe they need to see broken DNA to bind the DNA. So we thought, okay, maybe SIR6 is a double-strand break sensor. And that's why also the phenotype is so strong, and also all the repair mechanism is impaired. And to be a sensor, it has to recognize and bind double-strand breaks and initiate the DNA damage response. So what we did, first of all, we measured DNA binding. And we measured by EMSA. Do you know this assay? All the biochemistry is lost in you? Okay. So what we do is we label DNA with radioactive material, and then we can run it in a gel. If the DNA is bound to something, it will run slower. So free DNA is here in the bottom, and bound DNA is here in the top, because it's delaying its mobility. So we can see that SIR6 can bind very nicely single-strand DNA, and it binds double-strand DNA with a lag. It takes it a little bit longer, but then it binds more tightly, telling us that there is some cooperativity in there. So the model we came up is that we have for each strand of single-strand DNA a molecule of SIR6 that can bind it. But if we have a double-strand DNA that is broken in the ends, then we'll have some cooperativity, allowing the binding of two different molecules of SIR6. And in this way, they can cooperate and also keep together the DNA. If the DNA is broken and it starts kind of breathing, then we can start interacting with other things. But if we have two molecules that can bind it in the end, we will not allow this flexibility. And again, to confirm that this was forming a dimer, we put LAC SIR6 with GFP, so it's tightly bound in this place, and we put soluble SIR6 with RFP, and we can see it arrives to the sites where their SIR6 was, telling us that they can interact together. So these are some of the molecular techniques that we can use to measure signal in the cell. But in the EMSA assay, usually what happens is that we use always broken DNA, only small fragments of DNA that are used because they have to run in the gel. So we don't know if this is because it binds sequence specific or if it binds broken DNA. And to separate between these two, we develop an assay where we can take the same plasmid, we cut it with restriction enzymes. So in this case, we have exactly the same sequence, but cut and are uncut. And you can see here that MR11, that is the other DNA recognizing sensor, and CIR6 both bind the linear DNA, but not the circular DNA. This is in log. So we have very nice affinity only for broken DNA. We have exactly the same sequence as the circular DNA, but it doesn't bind. And we can have a little bit more binding when the DNA is in sticky ends, because maybe it's already breeding to separate and bind each one of them. And it's NAD independent, that this is the cofactor of CIR6 for deacetylation, for the epigenetic modification. So this allows us to understand that CIR6 really binds broken DNA. And then we wanted to understand how it can bind broken DNA. This is not a DNA binding molecule. We have done hundreds of assays to see if it binds specific promoters, and it doesn't do that. But when we analyze the structure of CIR6, we can see a region in here that has prediction, according to the amino acid characteristics, to bind DNA very weakly, but still bind DNA. And next to this region, we saw a ring. And in this ring, it can fit only a single strand DNA. So first, we check if this region that is in the core domain can bind DNA, and it can bind DNA nicely. And then we mutated all of these amino acids here in red that are in this ring to see if we can impair the binding to single-strand DNA. And again, we can see that if we mutate these regions, we have impaired binding to the DNA. So I know this is very biochemical, very deep into the repair system, but I also want you to know the methods that can be used to do the research. So please ask me questions if the methods are not clear, because maybe all the story won't be so important for you in the future, but how we can do the research and what are the important controls, this is what is important for me, for you to remember. So one of the ones that was the most affected, this HY mutant, is also next to the catalytic domain. And when we do laser-induced damage, we can see that really it cannot arrive to the sites of damage. If we induce laser-induced damage, wild-type CIR6 can come, but this mutant that cannot bind DNA, it doesn't come. So in this way, we have to use in molecular biology several techniques, because each technique is biased in a different way. Some of them don't allow me to see this, and some of them are biased by that. By combining different methods, such as EMSA plus the binding assay of the plasmid plus recruitment to the sites of damage, I can come up with a full story. Because if not, it's very fragmented. The pieces of information that we get are fragmented, and we have to try to put it together. So usually I tell the molecular biology is like being a cellular detective. You have some information here and there, and you have to build up a story with all these little pieces of information, so we have to fill the gaps as much as we can. Since we also saw that this was similar to the core domain, we measured the core domain of other sirtuins that are conserved, and we saw that some of them can also bind, but not as effectively as CIR6 binds. So again, we discover a new domain that can bind only broken DNA and not circular DNA. So to be a double-strand break sensor, we need two things. One of them, we have to recognize and bind the damage. And second, we have to initiate a DNA damage response. Because just by binding the DNA, it doesn't help us repair the DNA. So we have to be able to send this signal downstream. And for this, we again took advantage of this fake DNA damage. Why do we do that? Because when we do irradiation, many things are happening in the cell at the same time. We have hundreds of sites of damage. We have many molecules moving from one side to the other. We cannot say which one came first. We have to separate this, and for this, we can induce this fake damage. And in this case, we put lac CIR6 with GFP, and we see the lac CIR6 allows the phosphorylation of H2AX. And this is telling us that this is not a protein-protein interaction. This is an activated kinase. So this is telling us that we are really activating the downstream signaling. You understand the difference? If I just bring, for example, BRCA to the sites, This could be because these proteins interact together. So this is not telling me that I am activating the DNA damage response, but by having a phosphorylation, I am for certain activating the kinase that needs to do this phosphorylation. So we can see again that if we induce a fake damage with CIR6 or with the HY or MR11 as a control, we in all of them can phosphorylate damage to AX. Now I just told you that the HY could not bind DNA. So do you know why the SA still works in here? Any idea of why, if this one cannot bind the DNA, we still have activation? What? No, in what?
I don't know if it's correct, but I would just say that we have other pathways and other sensors to activate it, to phosphorylate the histone.
Well, in this case, we are activating by HY because it has lack. It doesn't require its own binding capability. We are faking the system and we are hijacking this capacity to bind the DNA. By itself, it wouldn't activate because it cannot bind. But now we are cheating the system. And this is telling us also that we need the core domain for the binding of the DNA, but we don't need it for the activation of downstream signaling. So to prove that, we use LAC with the full length or only the core domain. And the core domain, that is the one that binds the DNA, cannot fully activate the DNA damage response. So we need the C terminus and the N terminus for the full activation, but we don't need them for the initiation, for the binding. So it has different domains that can have different roles. And again, we check the other sirtuins, and some of them again have the capacity to activate the DNA damage response if they have the LAC domain. So we have a full new family of sensors that can identify broken DNA and send the signaling, and this is why maybe they are so important for the aging mechanism. This, when we have mutations in any of the sirtuins, we can have heart defects and metabolic defects and many other kind of age-related problems. So we think it can be related also to DNA repair. And last, we check if we have two main double-strand pathways for repair, homologous recombination and non-homologous enjoining. In here, the sensor is the mRNA complex, and in here, the Q complex. Where CIR6 fits in this story? And for this, we again use this pathway analysis with the proteins from non-homologous enjoining and homologous recombination, and we see that CIR6 can bring all of them from both sides. So the decision of which pathway we should use, it comes later. CIR6 is earlier than that, arriving really, really fast, but still allowing flexibility to which pathway we could send the cell. And you don't see this very much, but again, we can do laser-induced damage. And then we look for these lines that we generated with the laser. Imagine that we have the microscope and we come with the laser and we just draw things. You can do a smiley, you can do whatever you want. When you are bored enough by hours of doing this, you write your name and everything else. But then you do immunofluorescence, and we can see that control cells have gamma H2AX and RPA, that is the single-strand binding protein, and 53BP1 and BRCA1. I think the resolution is very bad with the red and green. But if we don't have CIR6, we lose the capacity to recruit these proteins to the sites of damage. So we don't activate the full response if we don't have CIR6. And we don't activate the full response in both pathways, homologous recombination and non-homologous enjoining. So we are impairing things from the very top and affecting all the repair. And again, just to see this is signaling and not only interaction, what we do, we prevent phosphorylation with wordmanin. Wordmanin prevents the kinases from working, so even if they are recruited, they cannot phosphorylate H2AX. And what we see is reduction in H2X phosphorylation, and also a nice reduction in the recruitment of 53BP1 and BRCA1. BRCA1, we see that they have also a direct interaction, but still by not activating the full system, we don't bring them as much as we wanted. So what we see in my lab that is important for SIR6 is that it's a double-strand break sensor. Until now, for more than 30 years, it was known that we have 3 sensors, mRNA complex, Q complex, and PARP. Now we have brought a new sensor that we really also see the phenotype, but we'll see later on in other classes, with cancer and neurodegeneration and heart disease, and it's affecting every tissue differently depending on if other partners that are present. We know that it arrives by 5 seconds independently of any other sensor. It can recognize and bind the double-strand breaks with no intermediates. We measure this with EMSA and with the binding assay, initiate the DNA damage response, and recruit proteins from both pathways. So now one of the projects in my lab is to try to understand how we decide if this is based in the epigenetic region. If we arrive to a gene-rich sequence, maybe we want to go to one pathway and not the other because we want to prevent mutations. Or maybe we have post-translational modifications done to SIR6 that can affect the recruitment of different proteins. And one of the things that we want to understand, that this is more dynamic changes, is what are the different proteins that come to the sites of damage and send the signaling, and what happens in the future. For example, if we have a double-strand break 20 years ago, can it still be marked epigenetically, telling the cell, I was damaged once, don't use me? So this is one of the big questions that we try to understand, because maybe when we accumulate too many of these epigenetic scars, this is the threshold to start a degenerative process. Like, okay, if we have too many of this, we should go to apoptosis. Or don't use this sequence. Or for example, we want to use this technology also in the usage of iPS. When we take cells from our cells and induce them to be pluripotent cells to put them back, for example, in our brain, we are already full of DNA damage, right? This is one of the barriers that we don't know what we're putting back. And you cannot all the time sequence them and find everything that is wrong. So what we can do is to test for epigenetic scars. If they have too many epigenetic scars, we could say, okay, we should not use this iPS in humans. We should go for the ones that have less this kind of scars. So these are some of the things that we are trying to find out in the lab. And just to summarize and finish, we have in the double-strand break repair several things that can affect the pathway choice. So these are one of the main active open questions in the field. How do we choose? Is the sensor that arrived can influence the outcome? Can it be kicked out if necessary? A part of the cell cycle, what are the other things that can influence how we are going to repair? And which are the proteins that are involved? Because one of the other things that may be happening is that once that we have damage, for example, and this connects us again to the epigenetic code. We have our DNA, and we have some modifications that were important for this region. But then we have damage, and we have to remove all of these modifications and put new modifications, like this GAMH2AX and other modifications that are important for DNA repair. But once the damage is done and we repair, did we bring back these same modifications as they were? Did we put epigenetic marks that are telling us this region is damaged? We don't know, and we think that part of the epigenetic drift that we see in aging is because after damage, we don't manage to put back everything as it was. So part of what we're trying to understand is all the restoration system, who they are, when they come, and if they manage to restore our DNA as it was before. So in the end, we have our DNA damage that when accumulates, it can send to signaling that when it's too much, we have genomic instability and accelerated aging. And when we have a good, healthy repair system, we have a good health span and we don't have too much tumors, and we are overall healthy. If we have inherited mutations in the repair system, we will have very early on cancer, or we will have premature aging diseases. We will see both these progerias and cancer, like well, several types of cancer. And if we have only mutation in downstream signaling, then we may develop cancer at early adulthood, in age 30, 35, as we gain the second hit. So that's why DNA repair is one of the most important processes in the cell, and we have to understand it better also to try to help it. So one of the things that personalized medicine is trying to do is, for example, where is what is here? Sorry. For example, if we know that a tumor has a mutation in non-homologous end joining, but the surrounding cells are okay, then we block in this tumor the homologous recombination. And in this way, these cells have no way of repair and are sending the cells to apoptosis, but the surrounding cells in the organism still have the non-homologous end joining. And in this way, we can reduce the dosage of all the chemotherapy that is very toxic to all our body, because we can target it specifically to the pathway that we want. So these are some of the things that we're trying to understand, the synergy between the different pathways of repair to allow a more personalized and less damage to the whole organism and the whole body. So that's it. Are there questions? You are always so quiet. Yeah.
No, I was trying to think about these other mammals that, for example, don't have high rates of cancer. What can we learn from them? Because, for example, I have heard capybaras or other mammals that, well, they are virtually free of cancer. So, and why do we have a lot of cancer as humans? I don't know if you maybe have an idea from your research or something.
Yeah, so many people are doing this comparative evolution, for example, also in naked mole rats. I didn't know about the capybara, it's called, but the naked mole rats, they have very low incidence of cancer, and they have a more robust DNA repair mechanism. And there was a very nice paper recently in Cell that they took CIR6 and they compared it from mouse to other animals the same size, other rodents, and they saw that the beaver, for example, can live 22 years, and a mouse lives usually 2 years. And they measure with the single changes in CIR6 the repair capacity. And they saw that the ones that live longer have a much nicer repair capacity. The enzyme is much more active and more efficient in DNA repair in the long-lived ones than in the small and short-lived ones. And then they used cells and changed the CIR6 to the one that was more efficient, and they became more resistant to many things that are toxic. Of course, CIR6 is just one example. It's a very important enzyme. I work with it, but of course I don't mean it does everything in the world, but it's a very good system to study aging. So many of the long-lived animals may have less chances of getting damage. For example, some of them live underground, so they don't have to develop such a strong UV-related response or have a lower metabolism. This was one of the things that was very important in this study, that they were all rodents the same size, because many theories about who lives longer came regarding our metabolism. How active is our metabolism and body size and all these things. And in the end, these theories were disregarded. It's like it doesn't influence how long do we live. But there was a very nice correlation with repair capacity for double-strand breaks particularly. So there is something in double-strand break repair, and also centenarians have a more robust DNA repair capacity than the main population, with less oxidative damage. That is something that is easy to measure, because some other types of damage are more difficult to measure. But there is a correlation with living longer and having a better repair capacity with some polymorphisms. No? Yes, sure.
Okay, so as the cells keep on living, you receive this DNA damage and you try to repair, and sometimes you cannot, and eventually this leads to all those problems. My question, which I think it does not have an answer, but I would like to see you talking about it a little bit, is that, okay, we have a lifespan and the cells can only live to a such point because of, in part because of those things. But how the living beings manage to like reset this thing in reproduction? Because we are like, the life is 2-3 billion years old and we're still reproducing and living, and there is something that makes these things not happen in the germinative cells in some way, that we can reset the thing and reproduce. So which are the things that could be important, and how does this work to a certain sense?
So one of the things that we know is that the germ cells are super sensitive to DNA damage. They won't go to repair mechanism, or they will try to repair just a little bit. They will go to apoptosis. Why? Because it's too much to handle, right? And all the life depends on them. They accumulate mutations during, like when the cells are replicating, but they have less damage or less capacity to repair the damage, and they and these are like the cheaters in the system, right? Like the germ cells usually are the cheaters of the system. Everybody stop dividing, differentiate, and help for the whole organisms, but the germ cells are cheaters. They take care only of themselves. And by doing that, like I think in evolution, this is the term, right? Why some populations evolve, because there are always some selfish population that takes care of themselves. So in a full organism, we call the germ cells cheaters. And they have almost no capacity to continue to reproduce with DNA damage. And we will have like a lot of miscarriage when we have people that is infertile. Many times it's because they have accumulated damage, or when the cell division is not complete, we can have, as in old age, more trisomies and other kind of diseases that are very dangerous. But we have very low tolerance for this kind of damage in germ cells. So this allows us to continue. In other cells, they will let it be until a certain point. So one of the things that we think, one of the theories of aging is this pleiotropic antagonism. Did you hear about this term? This is something that in evolution is good for us and give us fitness when we are young, but as we grow older, it not only doesn't benefit us, it causes more damage. And we will see this in the cell cycle class regarding to senescence. So when the cells get too much damage, they can go to this stage that is called cellular senescence, where they say, I am not sick enough to die, but I also will work only half time because I am sick, and I will also be nagging all my neighbors, sending signals, that pro-inflammatory signals that I am sick, I am not okay. So this, of course, is good because this alerts the immune system and prevents cancer, right? It prevents from these cells that are damaged to replicate. But when we accumulate too many of these cells, then they start destroying the system because they are nagging all of the neighboring cells. And this is one of the examples. So we have some things that give us fitness, but some things that we don't tolerate in evolution. So damage is one of the things that if you don't repair it, you don't continue dividing. This activates the checkpoints, and that's it. You are gone. And in stem cells, and even in germ cells, and even in stem cells, the capacity of repair is lower because you don't want to repair. You want to get rid of it. Okay? More questions? Yeah, sure.
I have a question about the use of all these DNA damage machinery, repair machinery by some other organisms like viruses. And you have the nuclear viruses, because all these things like a double break, double strand break, or a single strand break, they could be read by, like sensors like the sting and another type of sensors that we have inside the nucleus, and it will bring a immune response.
Right.
But then if we have like RPA that will cover this single strand, it could maybe be used by the own virus to to escape the immune system. So I don't know if you have something.
Like outside the cell, like the broken DNA that is secreted, or inside the cell?
No, inside the cell.
So inside the cell we have also many receptors, like TLR receptors, right? That when they recognize cytoplasmic DNA, they are activated, alerting the immune system that something is not right.
Mm-hmm.
Many of the viruses that co-evolve with us, they try to stop the TLR system and try to get into the nucleus to integrate in the genome,
Yep.
right? And they generate damage. So also one of the things that we're starting to think is that, for example, neurodegeneration, or now with the COVID, with all the symptoms that go to neurological and foggy brain and psychiatric diseases, that this activation of the immune system or this virus can affect the cells and can generate more damage, especially if they want to integrate or if they are constantly sending signal that this is nucleotides, RNA or DNA in the cytoplasm, telling the cells that something is wrong and activating the DNA damage response.
Mm-hmm.
So it affects not only inflammation, but also the intracellular mechanism to deal with it. And so in many cases, it can also send the cells to apoptosis or senescence when we have a viral infection. But depends on the virus and how much we know about it.
No, I was talking like because we have also these types of sensors for viruses, DNA or RNA inside the nucleus. And then like these sensors can be burled or escaped by this DDR machinery. So if we have something that is
like
already
like the viral retrotransposons and these things, or viral because I don't know.
The viral material itself.
Okay. I know that we try to block it, but I really haven't not read a lot about it, so I don't want to say anything that I don't know enough. But usually when we have, even when we cut in nucleotide excision repair a small piece of DNA that is in the genome that has to be degraded, it has to be coated to prevent activation of the DNA damage signaling.
Mm-hmm.
So I guess it will be similar, but at some point, if it's too much, it escapes the signal. But I really not sure about that.
Okay, thank you.
Okay. Any more questions? No? Okay.
So we are talking about a lot about aging, and it certainly has to do with DNA damage, but there is a kind of consensus, a definition of what's aging.
So aging is the slow deterioration of an organismal function until death. This is the definition. Now, in the aging field, the most accepted theory that why do we age is that we accumulate DNA damage, and the DNA damage signaling will start causing epigenetic drift, loss of proteostasis, mitochondrial damage can also be generated by DNA damage to mitochondrial DNA. But like we have the drivers and then we have the hallmarks. So the main drivers are DNA damage that can include also telomere attrition, right? Because the open telomeres will be recognized as double strand break. Or double strand break signaling or DNA damage will start all these signals that will stop the cell division, will induce also inflammation if we have, like you were saying, cytoplasmic DNA, and will initiate all the cascade that we see downstream. We will have a last class on aging and the different theories of why do we age, but one of the most accepted drivers of aging is DNA damage. And we see that this DNA damage can affect things downstream that are very common, such as proteostasis loss, or mitochondrial function, or intracellular communication, or energy balance. Because it is so important for the cell to keep its DNA intact, When it's not intact, it starts sending signals everywhere that we have to deal with this. So this is a driver. Of course, we can have other drivers if we have, for example, mutations, for example, in tau protein. And this can give aging, particularly only in the brain, because it forms aggregates and affects many other things. But a complete loss of function in progeria will come mainly from affecting the nucleus and the DNA. And this has been shown in many, many different disease and animal models. So this is the main driver, and other things will come downstream from DNA damage accumulation.
So all the diseases that are called progeria, they have to do with DNA damage?
Yeah, they have genomic instability. For example, with the lamin, they have genomic instability because the nucleus is breaking and the DNA is not controlled. So it's not a DNA repair enzyme, but it's the nucleus that is not contained and induces collapse. In the cocaine syndrome, like in the nerve that we saw, we cannot repair, and then we have this premature aging of most of the tissue, and if we have the other one, we have cancer. But also cancer is considered an age-related disease because our chances simply increase as we age and if we are unlucky. For example, there was, like right now, 3 families that were found with inherited mutations in SIR6, and this is embryonic lethal, with microcephaly and with not full development of the immune system. And if you have only 1 copy, they have like a shortened lifespan. So it is one of the main drivers. Of course, when we age, everything changes from the mitochondria to the metabolism to the intercellular communication. But what is the driver? Why are these things changing? And the main theory is that the DNA damage signaling is causing it. Okay? Good. So, coffee for